Hello everyone. Today we are going to make you understand the immunology behind organ or graft rejection. So before we get into that part, let's see what is organ transplant. Transplantation is a term used to transfer of cells, tissues or organ known as graft from one place to another or from one individual to another. Alexis Carrel reported the first systemic study of transplantation in 1908. He interchanged both kidneys in a series of 9 cats. Some of those receiving kidneys from one other cats maintained urinary output for up to 25 days. Although all the cats eventually died, the experiment established that a transplant organ could carry out its normal function in the recipient. The first human kidney transplant attempted in 1935 by a Russian surgeon failed because there was a mismatch of blood types between donor and the recipient. This incompatibility caused almost immediate rejection of the kidney, and the patient died without establishing renal function. The rapid immune response experienced here is termed as hyperacute rejection, and it is mediated by antibodies and will be described later in this video. The first successful human kidney transplant, which was between identical twins, was accomplished in Boston in 1954.
Now, let's see the terms used for different types of transplant. First is autograft, in which self-tissue transferred from one body side to another in the same individual occur. Example is skin transplant. Second is isograft, in which tissue transferred between genetically identical individuals. In humans, is the case of monozygotic twins. Third is allograft, in which tissue transferred between genetically different members of the same species. And the last one is xenograft, in which tissue transferred between different species. Example is graft of baboon heart into the human. So in this video, we're going to discuss about allogenic graft rejection, that is the graft rejection process occurring in the same species. So there are two pathways of the rejection. First is the direct pathway, which is also called as T-cell mediated process, and the second pathway is called as indirect pathway, which is also known as antibody mediated process. So let's have a look at the
In this process, tissues that are antigenically similar are said to be histocompatible. Such tissues do not induce an immunological response that leads to tissue rejection. Tissues that display significant antigenic differences are histoincompatible and induce an immunological response that leads to tissue rejection. In this mechanism, by which the recipient's T cells recognize the donor's MHC molecule without the need of processing by APCs. Therefore, it is known as T cell mediated mechanism. Process can be divided into two parts: sensitization phase and effector phase. So first, in sensitization phase, the antigen reactive lymphocytes in the recipient's lymph nodes proliferate in response to the donor alloantigen. In this step, the CD4 plus cells and the CD8 plus cells must proliferate vigorously in response to the foreign antigen. The CD8 plus cells will recognized by the MHC1 molecule, which are present on the surface of the cell. Then they will induce a cytokine mediated response by TC cells in order to kill the graft cells, which are identified as foreign antigen, which will in turn create a cascade of reactions such as coagulation, inflammation, and thrombosis. In thrombosis, the artery gets clogged, which is providing blood to the graft, and this will eventually lead to graft ischemia due to the low supply of oxygen as the artery is clogged. Now, if the CD4 plus cells will be recognized by the MHC2 molecules, then it will cause a delayed type sensitivity reaction known as DTH reaction. In some organs and tissue grafts such as kidney, thymus, and pancreatic islets, a population of donors APCs called as passenger leukocytes has to migrate from graft of the regional lymph nodes. These passenger leukocytes are dendritic cells which express high level of MHC2 molecules because passenger leukocytes express the alloantigen MHC antigens of the donor graft. They are recognized as foreign and therefore can stimulate immune activation of T lymphocytes in the lymph nodes. Now, recognition of the alloantigens expressed on the cell of the graft induces vigorous T cell proliferation in the host. This will cause the secretion of IFN gamma, which is a soluble cytokine, and activates the macrophages to destroy the graft cells. The second phase, that is the effector phase, The recipient sensitized cells will mediate the cell destruction. The hallmark of graft rejection involving cell mediated reaction is an influx of T cells and macrophages into the graft. And IFN increases the MHC2 expression as well. The CD8 plus by cytokines and the CD4 plus by the macrophages. Now let's see the processes that are acceptance and rejection of graft. In the autograft acceptance process, the first image depicts the day 3 to 7, known as revascularization. That means the graft has been implanted and the blood vessels are trying to reach the supply sufficient blood and nutrition so that it can survive. The second picture depicts day 7 to 10 in the process known as healing. In this, the blood vessels have reached the graft and formed a connection, and in the last picture, the graft has been totally accepted. In the autograft rejection process, the first picture depicts the same process as revascularization. Here also the graft has been implanted, and the second picture, that is of day 7 to 10, the process of cellular infiltration. As we know, the blood vessels reach the graft, and now our immune cells comes into the picture as in the fourth picture. We can see that the blue colored cells that are neutrophils, and thus the artery is getting clogged so that there is no blood supply to the graft, and thus this process is known as thrombosis, and therefore the graft will go into necrosis. As depicted into the last picture, necrosis, as the graft cells are dying and thus the graft is getting rejected. So this was the gist of processes of acceptance and rejection of graft.
So now let's discuss about indirect pathway. So in this process, allorecognition by TS cells occurs, which is acquired by the host APCs and therefore presented by MSC class II molecules. Some antigens of phagocytous graft cell appear to enter MSC class I pathway presentation and are indirectly recognized by the TC cells. This phenomenon is called as cross presentation. It is known as cross presentation because the MSC class I pathway is going to get involved in it, and since the indirect pathway is supposed to be MSC class II pathway. Generally, this pathway is triggered by trauma from transplant itself and can occur during the procedure with release of foreign proteins from the graft cell that are taken up and further proceed by recipient APCs resulting from the pro-inflammatory environment. The recipient antigen presenting cell, that is your APCs, will have MSC class II molecules on them, which will recognize the CD4 plus cells, and the TS cells will be activated. Now, since TS cells are activated in this process, this will interactivate B lymphocyte, and we all know that B lymphocytes are mainly involved in antibody production, and so therefore this process is known as antibody mediated hypersensitivity reaction, because in this process antibodies are produced in abundance. Now this reaction will activate plasma cells and antibodies which are released in the graft cells. These antibodies, which are dispersed in the graft tissue or organ, will act as a flags of destruction for the graft tissue or graft cells. These antibodies will be recognized by the macrophages and cause endothelial injury, and in turn it will cause graft ischemia because the artery which is supplying blood to the graft is now blocked. Therefore, there is no supply of nutrition as well as oxygen to the graft. This will cause ischemic condition known as graft ischemia. This will eventually kill the graft cell and cause graft rejection. So as we can see that in this process, the major role is of antibodies which are produced by the B lymphocyte and are further destroyed by the
Direct and indirect processes. So in this picture, as we can see, the first is the APC cells, which is being recognized by the TH cells. Now the TH cells is categorized into four types of cells: the TDTH cells, the CD8 plus cells, the CD4 plus cells, and the B cells. Now the first three, that is TDTH cells and CD8 plus and CD4 plus cells, these come under the category of direct process, and the B cell is the indirect process. So let's see the gist of direct process. The first is TDTH cells, in which the IFN gamma and IFN beta are taken into action. The IFN gamma is going to activate macrophages, and thus macrophage is going to destroy the graft cells, and in second the IFN beta is going to induce a cytokine reaction on the membrane of the graft, therefore causing it to lyse the membrane of the graft, and thus the cell die. The second is CD8 plus cells, which is going to be recognized by MHC class 1 molecule, which is present on the surface of the graft. And the third one is CD4 plus cells. This is going to be recognized by MHC class 2 molecule. Both the MHC class 1 and MHC class 2 molecule, when being recognized by the CD8 plus and CD4 plus cells, are going to induce a membrane damage of the graft cells, thus causing the lysis of the graft cells.
So now, as you can see that in indirect pathway, the TS cells is going to activate the B lymphocyte cell. Further, this B lymphocyte cell will in turn produce antibodies which will go and sit on the surface of graft cell, and they are going to get recognized by either NK cells or macrophages. This recognition is done by the Fc receptor. Now this Fc receptor will recognize the antibodies which are present on the graft cells or graft tissues, and then these macrophages will kill the graft tissue. So this is how we came to know about the two pathways, direct pathway and indirect pathway. And by both the processes, graft can.
Let's talk about some facts now. There are instances in which an allograft may be accepted without the use of immunosuppressive measures. Obviously, in this case, the tissue that lacks alloantigen, such as cartilage or heart valves, there is no immunological barrier to transplantation. However, there are also instances in which a strong predicted response to an allograft does not occur at all. There are two general cases in which an allograft may be accepted. One is when cells or tissue are grafted to a so-called privileged site, that is sequestered from immune system surveillance. The second is when a state of tolerance has been induced biologically, usually by previous exposure to the antigens of the donor in manner that causes immune tolerance rather than sensitization in the recipient.
Now let's see challenges faced during organ transplant surgery. During organ transplant surgery, doctors are always racing against the clock, and the time is not on their side. Also, during the surgery, several precautionary and preventative measures are taken into consideration. But still, the major challenge is about keeping organs viable, that is alive outside the human body before being transplanted. Well, depending upon the organ, the time range can vary from few hours to day and a half. For example, liver has a viability of 12 hours, lungs 6 to 8 hours, kidneys 24 to 36 hours. These are the commonly transplanted organ. But when it comes to heart, it's not that easy because heart loses the viability the fastest when stored in the cooler. Also, we have only 6 hours to work with because the longer it is outside, more difficult it is to transplant. But the new medical industry known as Transmedic is making a difference. The company has developed an organ care system known as OCS. It is an integrated system that allows transplant teams to keep organs alive, beating, breathing between the donor and the recipient instead of storing them on ice. After the heart is removed from the donor, it is attached to what is called as organ care system or OCS. It kept it warm, it fed nutrition, it circulates the blood. Basically, it kept it alive. The OCS heart and OCS lung system are CE marked and in clinical use in transplant centers in Europe, Australia, and Canada. Hence, heart in a box is going to replace the old therapy and nothing to do with the.
Let's get to know some statistics as what is going on in the world of organ donation. So, every year, August 13 is celebrated as Organ Donation Day. The day is celebrated to motivate and encourage people to donate their organs and to understand the value of organ donation in the life of an individual who needs it, because lack of people die every year waiting for an organ, and every 12 minutes one patient is added to a waiting list. According to a survey, every year in India, about 500,000 people die because of non availability of organs. 200,000 people die due to liver disease. 50,000 people die because of heart disease. Moreover, 150,000 people await a kidney transplant, but only 5,000 amongst them get it. Despite India being the world's second most populous country, it has a pathetic diseased organ donation rate of just 0.26 per million population compared to 26 in US, 35 in Spain and 36.5 in Croatia. India is struggling with an acute shortage of organ transplantation. It is estimated that more than a million people suffer from end stage organ failure, but only a handful of 3,500 transplants are performed annually. Awareness of organ donation is therefore the only way to be out of this depressing scenario. The more potential donors there are, the more likelihood of the organ becoming available to save lives. Now, let's end this video on a positive thought.
The idea is not to live forever,
But maybe to help another live a little longer. Thank you, everyone.